Hello and welcome. Inside our bodies right now, there's a process happening trillions of times a second that's absolutely fundamental to life. Today, we're going to follow the incredible journey of a protein, seeing how a simple instruction from our genes becomes a complex working machine. Let's dive in.
So, to get us started, think about this for a second. We've got chemical messengers for your brain, tiny soldiers for your immune system, and the very stuff that lets you move. I mean, they seem completely different, right? What could possibly tie them all together? Yeah, guessed it. The answer is proteins. They are, without a doubt, the hardest working molecules in our cells, doing just about everything. And today, we're going to follow the life story of one of these amazing molecules, from the moment it's conceived to the moment it gets to work. All right, so to build anything, you need three things: a plan, a factory, and the right materials. Well, it's exactly the same in the tiny world of a cell. So let's meet the key players that are responsible for building every single protein. First up, we've got the blueprint. This is messenger RNA, or mRNA for short. Now, you can think of your DNA as this giant, priceless cookbook locked away in the cell's nucleus. The mRNA is like a quick photocopy of just one recipe. This copy gets sent out of the nucleus to the main factory floor, which we call the cytoplasm. And here's the factory itself, the ribosome. And this thing is an absolute marvel of natural engineering. It's this huge machine built from its own special RNA and proteins. Its one and only job? To grab that mRNA blueprint and start building. And of course, every factory needs a supply chain. That's where transfer RNA, or tRNA, comes in. These guys are like little delivery trucks. They zoom around the cell, find the exact amino acid building blocks we need, and haul them over to the ribosome right when they're called for. Okay, so we've met the team, we have the blueprint, the factory, and the delivery trucks all ready to go. Now, let's watch this incredible assembly line, a process we call translation, spring into action. Translation basically happens in three main stages. First is initiation. That's where the ribosome finds the start sign on the mRNA and clamps down. Then we get to the main event, elongation. The ribosome chugs along the mRNA blueprint, reading the code and linking those amino acids together, one by one, into a long chain. Finally, we hit termination. The ribosome sees a stop signal, the whole complex falls apart, and voila, the brand new protein is set free. You know, it's pretty fascinating when you stop and think about it. RNA is doing everything here. We have mRNA as the message, tRNA as the carrier, and even ribosomal RNA as the heart of the factory itself. This deep involvement of RNA is actually a key piece of evidence for what's known as the RNA world theory, this idea that very early life on Earth might have been based entirely on RNA, long before DNA and proteins ever came along. So, there we have it. A new protein chain has been built. It exists, but it's just kind of floating there. Its job might be across the cell, or maybe it needs to be shipped out of the cell entirely. So believe it or not, its journey is really just getting started. So how does that happen? Well, the cell has this incredibly sophisticated system, basically a cellular postal service, to make sure every single protein gets delivered to its proper workplace. This whole process is called protein sorting. Now, the cell uses two main shipping strategies. First, there's the secretory pathway. This one is for proteins that need to be embedded in membranes or shipped out of the cell, and it actually starts while the protein is still being built. The other strategy, the uptake targeting pathway, is different. It waits until the protein is completely finished and then delivers it directly to its final home, like the mitochondria or the nucleus. For this explainer, we're going to follow the busiest route, the real superhighway of the cell, the secretory pathway. This is the path taken by super important proteins like insulin, or the receptors that sit on the surface of your cells. So as the protein starts to get made, a special sequence right at the beginning acts like a shipping label. This label directs the entire ribosome over to the surface of the endoplasmic reticulum, or ER. The new protein is then threaded inside, and this is where the quality control starts. It gets help folding into the right 3D shape, special bonds are formed to make it more stable, and, this is super important, complex sugar chains are attached. That process is called glycosylation. From the ER, our protein is off to the next stop, the Golgi apparatus. Now, if the ER was the workshop for quality control, the Golgi is the central post office. It's the packaging and shipping center. Here, proteins get a few final tweaks, those sugar tags can be changed a bit, and then they're sorted and packed into little membrane bubbles called vesicles, all ready for final delivery. And now we're on the final leg of the journey. These little vesicles start budding off from the Golgi, kind of like a whole fleet of tiny specialized delivery vans, and each one is addressed to a very specific location. Some of these vesicles will head straight for the cell's outer membrane, either to become part of that membrane or to release their contents immediately, like with collagen. Others are part of a regulated pathway, meaning they're stored and only release their cargo, like insulin, when they get a specific signal. And then there's a really cool one. Proteins headed for the lysosome, the cell's recycling center, get a special sugar tag, MSP, which acts as a unique molecular zip code, ensuring they go to the right place and nowhere else. So you might be thinking, this microscopic assembly line is fascinating, but why does it matter so much? Well, it turns out this process has massive implications for our health and for the future of medicine. Let's take a look. Okay, let's look at a really clear example. Remember those sugar tags that get attached in the ER and Golgi? Well, the exact structure of those sugars can actually be different between species. This chart, for example, shows how human cells tend to put a specific NANA-GAL unit on the end of their sugar chains, while pig cells use a GAL-GAL unit. It seems like a tiny, tiny difference, but in medicine, it's a huge deal. That one little difference in a sugar tag is a major hurdle in xenotransplantation. That's animal-to-human transplants. Our immune system sees that wrong pig sugar, recognizes it as foreign, and launches an all-out attack. Now, a lot of cutting-edge research is focused on this. Evidence suggests that if we could genetically engineer pigs to have more human-like sugar tags, it could be a path to solving organ shortages. But it's an incredibly complex problem, and current research indicates the technology is just not yet ready for safe clinical use. So let's end with this thought. This whole unbelievably complex process, from blueprint to factory to final delivery, is performed with breathtaking precision trillions of times a day inside you. But what happens when there's just one tiny mistake? When a single genetic typo leads to a protein that can't fold right, or an address label that's written incorrectly? Well, that one tiny error is very often the root cause of so many genetic diseases. It's a powerful reminder of just how elegant and how fragile this factory of life really is.